During this meeting, more or less in each presentation, we saw one of those pictures. And it's normal because our meeting is about His Majesty chromatin. The problem of all those pictures is that they are not full. They are not full because, let me remind you what exactly is the chromatin. By very simple definition, is a complex of DNA and proteins that comprise eukaryotic chromosomes. Well, the proteins are classified in two big groups, histones, which I won't talk about, and non-histone proteins, which are divided by two groups, the functional one, which are involved in gene regulation, and the structural one. In this talk, I will focus our attention on the proteins which are involved in chromosome structure. So high mobility group proteins were discovered like maybe 50 years ago, and they were described by those specific properties that I listed here. Since then, scientists are how to say, in the beginning, they studied them a lot, and especially for HMGB1, I can tell you that this protein was found to participate in practically all DNA-dependent processes. It was described to participate in replication, repair, chromatin organization, transcription, in everything. I want to point something, to stress on something. HMGB1 is very abundant protein. Normally, in normal eukaryotic cells, you have one molecule of HMGB1 at each 10 nucleosomes. Why? We have to keep this in mind. So, a little bit about the structure of the protein. It's consists of two boxes where the modern name of the protein comes, high mobility group box protein. Those boxes are the active part of the molecule by which HMGB1 interact with DNA. And in the end of the molecule, there is a acidic stretch, which is very important for a lot of the functions of the protein. So here I'm going to show you the structure of the two boxes, which are the active parts of the protein, the crystallographic structure, which was discovered by Jean Thomas and Andrew Travis, and they showed that protein could exist in a more close or more open conformation. And nice crystallographic work came up recently. The authors showed that actually for DNA binding, two molecules of HMGB1 are needed, and actually during this interaction, the phenylalanines are intercalate in the DNA bases. Okay, since in the beginning, after the discovery of HMGB1, all the scientists who were studying HMGB1 were concentrated onto the role of HMGB1 in the nucleus. Nowadays, since it was discovered that HMGB1 could act as a single molecule in certain circumstances, nowadays all the scientists, almost all the scientists who are dealing with HMGB1 are concentrated on this part of the functions of the protein as a signal molecule. They are very, very interesting functions, of course, and even in my lab we switched and started to study HMGB1 in terms of cell migration and proliferation, differentiation, apoptosis, and cancer. But this talk would be focused on this part. In my lab, we studied HMGB1 in terms of replication, repair, and nucleosome sliding and remodeling. So, a little cartoon to show how HMGB1 interact with the DNA, the bands. Okay. In the term of chromatin, it is, as I told you, HMGB1 acts as an architectural factor. So in the term of chromatin, what we imagine that happens, for instance, for initiation of transcription, or let's say, transcription, let's imagine that transcription factor is there, but as the initiation sequences is wrapped around the nucleosome, something is needed to happen in order to start transcription. So let's assume that HMGB1 came, it kinked DNA and pulled it out from the nucleosome, and this partial unwrapping creates the possibility for transcription factor to recognize its sequences and to start the transcription. So my career began with studying post-synthetic modification of HMGB1. So I'm tempted to show you some of those results. So this picture is from my PhD thesis. In our lab, we have methods to obtain in vivo acetylated HMGB1 protein and to study how this modification influences the role of HMGB1. So we started with, as I told you, HMGB1 has a great affinity to distorted DNA structures as cisplattinated DNA, four-way junctions DNA, or UV damaged DNA. And we discovered that post-synthetic acetylation of HMGB1 increased the affinity of the protein to all those structures. We quantify the KDs of those interactions, and you could see that KDs for acetylation protein are much lower than those for normal protein. So our hypothesis is that a possible mechanism of behind this increased affinity is in some interactions between acidic C-tail and the lysine in second position, which is subject of acetylation. If you remember the model of Jean Thomas, they showed that the protein could exist in an open and closed conformation, and we think that this acetylation favor the open conformation of the protein. That is why it's more reactable. And here is a little cartoon expressing this. So our next question was how post-synthetic acetylation influenced the possibility of protein to interact with linear DNA. And we were happy to understand, to see that there is a big difference actually. The normal protein, not modified protein, when interact with a short piece of DNA, like less than 100 base pairs, we are, I believe that, you know that DNA is kind of rigid molecule, and if you have a small pieces of DNA, you could not get it in circle in the presence of light gases because it's kind of rigid. It could be happen only in the presence of a protein who could kink DNA. And HMGB1 is one of those proteins. So here we used 97 BP piece of DNA, and in the presence of light gases, we obtained those circles, which increase in a concentration-dependent manner in the presence of non-modified protein. But when we put acetylated one, we didn't see any circle. Instead, we obtained those dimers and trimers, which was kind of okay. It could be happen if somehow acetylated protein has a great affinity to the DNA ends, and this somehow make ligation more possible. So again, we have this model for circling and for end joining. This is just our hypothesis. Next, we did some atomic force microscopy, and indeed, it turns out here is a linear DNA, here is DNA in the presence of HMGB1, and you could see that indeed non-modified protein has a great affinity to the center of the DNA fragment, and acetylated protein binds mainly in the ends. And here we also find a lot of this kind of structures. Those are two fragments bringing together by this by acetylated protein. We scored this all these complexes, and indeed, as you could see, regular protein has a greater affinity to the internal binding, and acetylated one has greater affinity to the ends. So another results are connected with the role of HMGB1 in nucleosomal sliding. Here we used a DNA fragment with positioning sequences. These are special DNA fragments in which there are specific sequence which create a curvature which favor the nucleosomal formation. And as you know, in the presence of remodelers like Swiss NIF, you could push the nucleosomal octamer from center, as it's shown in this case, to the end. And this could be observed by EMSA. So we did this experiment in the presence of HMGB1, and it turns out that HMGB1 could influence this process, and acetylated form is much more active. We did a lot of experiments, but here is the main results, which is actually that in the presence of HMGB1, it stabilizes the binding of Swiss NIF complex to the DNA. That was our conclusion of all these experiments. HMGB1 by structure is a wonderful candidate to be chaperone molecules because it has positively charged boxes and negatively charged acidic tails, which are characteristics of chaperone molecules. So next questions was if HMGB1 could help nucleosomal formation. First, we tested the role of the protein in classical nucleosomal formation by salt dialysis, and it turns out that indeed HMGB1 was increased the nucleosomal formation. Then we tested next possibility, HMGB1 to help nucleosomal formation just in the bench without salt dialysis, which is impossible if in the tube you don't have some architectural factor to help this process. Otherwise, you couldn't get nucleosomes, which is shown here. And indeed, in the presence of HMGB1 proteins, we obtain nucleosomes. And we did the experiment twice in two different versions. First, we incubate HMGB1 with DNA, and we got these results. And then the other way was to incubate HMGB1 with histone octamers to see if this is the important interaction, but it turns out that no. So HMGB1 could be a DNA chaperone. Okay. So the conclusions of this is that acetylated HMGB1 acquired the property to form a stable complex with the core particles. Okay, I won't listen this. Instead, I want to show you some very, I'm in the end, to show you very recent results which were inspired by the work of Stoinu and the wonderful approach that he developed in his lab. As I told you, HMGB1 recognizes a lot of DNA structures like UV damaged DNA or cisplatinated DNA or this cruciform DNA. So we wanted to test how HMGB1 interact with dynamically with DNA lasers caused by UV laser. So here I want to show you our results. You remember this figure from the presentations of Stoinu and all those clustering of different proteins which are involved in repair. And we found HMGB1 here, which means that HMGB1 is one of the first proteins who recognize DNA damage. And then he went out of the damage, and now we want to study these mechanisms and what could be the role of HMGB1. We want to find if it interacts with somebody of those guys. So this is a new project that we focused on. And now to go to the end of my talk, as I told you in the beginning, nowadays almost all the scientists who are dealing with HMGB1 actually studying the role of the protein as signal molecule and extracellularly. But do not forget that HMGB1 is nuclear protein, so it's there for a reason, and these reasons is not discovered yet. And you guys, I think, already have a lot of approaches to ask these questions. And actually, my talk has the goal to inspire you. No, because, okay, motivation, yes, we know what is motivation. We have a lot of work to do. But my talk, the goal of my talk is to inspire you and to remind you that HMG is waiting for you. Let's discover the role of the protein. And here are the people in my lab, and here are the funding bodies, and thank you for your attention.
Ok, Okay, I don't have a question, just small comments. I think many of you probably here know Bob Roder. Bob Roder was the first person that did transcription of chromatin in cellular extracted, in vitro transcription. And he tried to do it by purified factors. So it's a monster work. People, he has more than 30 postdocs at that time, or maybe 40. People purified all the factors, all transcription factors, general transcription, all the transcriptional component, no transcription. Finally, they start, fractionate extract, and they check all the components to add it to this soup, okay? And finally, they get transcription. You know what's the protein they found that induced transcription?
Of course I know, HMGB2.
Yes. So.
Any other questions? If not, let's thank Iva and the rest of the speakers.
Thank you very much.
We'll be back at 10:15. 10:15?
Oh, such a...
Yeah.